Morning to all. Good morning to all. Dear delegates.
Can you listen to me, Rajan?
Yeah, ma'am.
Okay.
Ma'am, Lakshmi ma'am,
Yes.
Tell me.
am I audible, ma'am?
Yes,
Yeah.
you are audible.
Okay, thank you.
I am audible now.
Yeah,
Ma'am,
yeah.
am I audible, ma'am? Aarti here.
Yes,
Yes,
yes, yes, Ardi.
All of you are audible. Okay, all the best.
Am I audible? Am I audible?
Yeah, yes, sir.
Yes, sir.
You are audible.
Thank you.
You are starting.
We'll go on live in two minutes. Please start after 2 minutes.
Okay, fine.
Start, ma'am, please.
A very good morning to all. On behalf of Arupade Vidu Medical College and Hospital, Puducherry, and the organizing team of the BMSCON 2022, the 3-day International E-Conference on role of basic medical sciences in academia, diagnosis, and research advancement. I welcome you all to this conference. This is the third consecutive year that the preclinical departments of anatomy, physiology, and biochemistry, along with the Center for Biomedical Research, are organizing this virtual conference. A heartfelt thanks to everyone who has joined us today. The inaugural program is scheduled at 9:45. Without further ado, we start our conference. We are very happy to begin the conference with the keynote address by an illustrious speaker, Dr. Chandan Sen, and our distinguished chairpersons, Dr. Ravi Kumar Chittodia and Dr. Koshi are there, along with our moderators, Dr. Gopinath and Dr. Shushmita Mahapatra. I request Dr. Gopinath and Dr. Shushmita Mahapatra to take over the proceedings. Thank you all.
A very good morning. I'd be privileged and happy to introduce our first chairperson, Dr. Ravi Kumar Chittodia, Professor of Plastic Surgery, Registered Academic and Head of IT Wing and Telemedicine. He did his graduation MBBS and post-graduation MS General Surgery and MCH and PhD in Plastic Surgery in India, higher training in USA, and he has 9 fellowships, including fellowships from Royal College of Surgeons of Edinburgh. He is presently Nodal Officer of NMCN project and NKN project and ISRO project Telemedicine, Government of India. And to his professional experience and achievements, he has research and assistant editor of Journal of NGMC, current editor-in-chief of Journal of Plastic Surgery and Transplantation. He has authored 5 textbooks, 7 manuals, and almost 6 chapters and 278 publications to his credit and 1 patent. He is recipient of 31 awards, including BMJ International Award and SKOCH Award. We welcome you, sir, for starting this session.
Good morning. At the outset, I would like to thank the organizing team, including the organizing chairperson and the organizing secretary, and all the participants and the faculties. Good morning once again. It's a very interesting session to start with the keynote address from Dr. Sen. And we all know that regenerative medicine is the upcoming and the most promising when it comes to regenerate the tissues, especially those like burns, diabetic foot, necrotizing fasciitis, or any other raw area which needs to regenerate. And we also know that nanotechnology is existing. And also we are aware of tissue engineering. But when we combine all these 3 technologies, then we are coming up with the so-called nanotransfection. And our speaker is the pioneer and the recipient of award and one of the authority, I can say, And currently he is the director of Indiana Center for Regenerative Medicine and Engineering, and associate president, G.S. chair and professor of surgery, executive director, Indiana University Health Comprehensive Wound Care Center, and vice president of research, surgery, associate dean, School of Medicine, Indianapolis, Indiana, USA. His credentials, awards, publication, H-index is innumerable because it's already 9:10. I should not take his time. So over to you, sir. We are eager to listen from you.
Very good morning to all of you. I would like access for sharing my screen, and somebody please help me with the process such that, let me see if I need to do the audio transfer. So let me check. All right. I suppose you can see the slide. Just swap it here. All right. A very good morning, and many thanks to the organizers for having me. And I have my Science College fellow student, Dr. Sushmita Mahapatra, with you, and thanks to her for bringing me here. Greetings to my co-panelists. Greeting to all in the audience. And I'm going to share with you, these are very precious moments that we will spend together the next, say, 40 minutes. And I'll try to communicate some message that sticks with you. I'll not get into the nitty-gritty complexities of science. This being a keynote, I'd like the scale of this presentation to be broad-reaching so that each one of you in the audience can connect with it. And hopefully we'll have an active discussion session if we have time for it. Thank you again. So today I'm going to talk to you about regenerative medicine. And so it's a new discipline in modern medicine. And speaking of modern medicine and the implementation of modern medicine, much of modern medicine, all of modern medicine has to travel through a regulatory path. And if we look at the history of such regulatory path, it was in 1906 that the FDA was founded in the US. It was in 1940 that the Drugs and Cosmetics Act of the Government of India, formed the CDSCO. And the CDSCO is currently in FDA Bhaman. I was very recently meeting Dr. Somani, the Director General of CDSCO, and I've been there several times. It's in Kotla Road in New Delhi. Now, before we get into regenerative medicine, it's important that we have a little bit of a context, historical context. So you see, in 1906, FDA was born. But at that time, it was primarily focused on regulating food material, so that food material is not adulterated, so on and so forth. Then came the revised Food, Drug, and Cosmetic Act in 1938. And it is at this time, in 1938, that primary focus was directed on the regulation of new drugs, and that people could not make false therapeutic claim in drug labeling, so on and so forth. So that was 1938. It was in 1982 that this was again revised to make room for what we know today as medical devices. So we had drugs, we have devices, and then something special happened in the US, FDA, in December 13, 2016. And what is that? We had the 21st Century Cures Act, as a result of which FDA, in addition to medical devices, have now added the RMAT designation, regenerative medicine and advanced therapy designation. So to make it simple, soon you will see human body parts made in the laboratory or elsewhere would be waiting to be plugged back into the human. So we had medical drugs, medical devices, and now we are entering into a new era of healthcare that would see the presence of, in one way or the other, human body parts that would be plugged into the body. Now, this is not about, it is not my field, it is your field. Anybody and everybody interested in healthcare must pay attention to this, because I just took a couple of clips from the news, one in November 14th, the other was published today, November 16th. And you will see that this is already a significant market, over $20 billion a year, and is supposed to be roughly $60 billion in the next 8 years. So if we don't pay attention to this, we will have other people advance with this and we will be followers. So it's very important, if you really want to participate in this growth, to actually take interest and actively participate while the field is developing. So you see, through the RMAT track of the FDA, until the end of 2021, there were 67 approved products. And this year so far, there are 12 more. So you have something like 79 products already approved through the RMAT track. Now, when you talk about regenerative medicine, people mostly think about stem cells. And stem cells, in my opinion, is one version of the existence of a cell where you have maximized plasticity. Now, unless we saw stem cell, we would never depart from our classical mindset of terminal differentiation. So, you know, tissues are terminally differentiated, and we always know that once they're terminally differentiated, they stay that way. They don't reverse course. But once we started knowing about stem cells, that remarkably changed. So now that story is like 2 decades old, and now stem cells, there's a lot of good biological research. However, the issue with stem cell is, if you have a pool of stem cells in your hand, and if you're injecting into the patient's body, some cells will become what you want them to become. But other cells may not listen to us and even become cancer cells, causing complications. So we are dealing with a lot of issues with stem cells. Now, that is an extreme form of cell plasticity. What I'm going to talk to you about is that you do not necessarily need stem cells. Pretty much every cell of the body has some level of plasticity. And if you pick the right tissue and you pick the right cell type, you would be able to actually change state or fate. I'm going to come to those definitions shortly, without actually using stem cells. So why am I right now not so interested in stem cells? Because stem cells are wonderful in trying to understand cell plasticity, and they are tremendous with respect to their potential. But today, we don't know enough to really safely handle them. So this article was authored by the FDA in the New England Journal of Medicine, where you see that the FDA is quite excited about this new track. However, as expected, they are laying a lot of emphasis on safety before we talk about efficacy. So safety first. Now let's go back to the concept a little bit. So you see, if you talk about tissue plasticity in vivo, we know that under fetal conditions, the tissues are highly plastic, meaning if you went in utero and you did some injury, when the baby is born, you can't even tell where the injury was made. The repair that happens in the mother's womb is near perfect, or as we call it, regenerative repair. But once the baby is born, and of course, when the baby then grows up to be an adult, this regenerative repair cannot be recapitulated. So in the child, you have blunted plasticity. And in the adult, as we learned in the textbook when I went to school, that the adult tissues are terminally differentiated, so they're not supposed to have plasticity. But if you now look at what we know today, in adult tissue, there can be 3 categories: low plasticity, intermediate plasticity, and high plasticity. So this concept of terminal differentiation is no longer valid. And if you now mess around or play around with adult tissue with high plasticity, for example, the skin, the fat, then you can actually generate cells and tissues of interest that could be of therapeutic benefit. Now, why have we been so excited about stem cells? Because stem cells actually taught us that plasticity can happen. And then when we knew plasticity can happen, we started looking for plasticity in cells other than stem cells. But in brief, stem cells can make more stem cells. Stem cells can make any one of the 206 specialized cells of the human body. But often what we don't talk about is that stem cells can also make cancer cells. So once we inject the cells into the human body, we are responsible not only for the cells that actually became the muscle or the nerve we wanted, but also of the cells that did not listen to us and had this type of a neoplastic fate. Now, most of you would have heard about induced pluripotency. In brief, originally fibroblasts from the skin were taken, and then you grow these fibroblasts in the petri dish in the laboratory conditions, and then you go through a process that is several weeks, could be easily 3 weeks, and you now generate pluripotent stem cells. So now these cells taken from the adult skin have been converted to IP cells, or induced pluripotent stem cells. Now, based on what you do to these cells, you could make beating myocyte, you could make gut cells, you could make blood cells, skin cells, neurons, you could make a whole lot of stuff. But all of this happens in the laboratory. So when you now take these cells, although those fibroblasts came from they were autologous, when you put them back into the body, the immune system is not happy. The immune system objects. So you are confronting with the problem. Plus, most of these pluripotent stem cells will not listen to us and go to the fate we want, and would continue to be stem cells. So if we now inject these cells into the human body, we have again the risk of stem cells becoming cancer cells. So embryonic stem cells, we know plenty about it. We just discussed about induced pluripotent stem cells. But notice at the bottom of your screen, and you will see an arrow highlighted in yellow saying direct conversion. Now, this concept of direct conversion, where an adult cell, so mind you, we had adult fibroblasts from the skin, we made them into pluripotent stem cells, and then we brought them back to, say, neuron or muscle or whatnot. But in this case of direct conversion, you do not need to take the fibroblast to stem cell. You can directly convert the fibroblast from a fibroblast into, say, a neuron, or into, say, an endothelial-like cell, or a muscle cell, so on and so forth. So this direct conversion is of interest because now you don't have any intermediary stem cell, so you don't have to worry about handling stem cells that don't listen to us. So fibroblasts, this process of direct conversion, if you failed, you are left with fibroblasts that you had to begin with. If you succeeded, you now have the cell you wanted. You don't have to deal with any intermediary stems. So what was our interest? Our interest was to develop a technology where the reprogramming does not happen in a laboratory. First of all, scalability of anything happening in a cGMP laboratory is very low. Only very few people will have access to it. It is highly expensive. Plus, I told you, if you do these changes in the laboratory, bring it into the intact body, you will have to fight the immune system. So we wanted to develop a technology where no laboratory is needed. We will actually do the reprogramming, not of cells, but the entire tissue. So the field of tissue reprogramming came forth. We are going to do it because we are doing it in the body. We are doing it under immune surveillance and in negotiation with the complex in vivo microenvironment. If you're doing it in the laboratory, of course, you can manipulate a number of parameters, but those parameters would never approach the complexity that you have in vivo. So you're going to be achieving this tissue reprogramming in constant negotiation with a complex and dynamic in vivo microenvironment. If we can achieve such technology, we felt that it would be giving us a sustainable functional impact. So this is the original version of the technique with the first time we did in the laboratory. So we developed this technology called TNT, or tissue nanotransfection technology. So this involves a nano chip, as you see the square sitting on the skin of the hand here. And then you have a couple of electrodes, and this entire process is 100 millisecond long, 0.1 second long. And then you only do it once. The chip does not stay with you. So you're done in 1/10 of a second, the chip goes away, and then this reprogramming starts. I'll show you then what happens. So when this was first published, the NICE director wrote a blog, and if you go to YouTube and write tissue nanotransfection, there are more than 1,000 videos talking about it. Different people are coming up with different ideas about it. So should you be interested, go to YouTube and see. And there are a few videos from our own group. I'll share with you a couple of them. Here is the first one. I hope you can hear the sound.
As cells differentiate, they acquire specialized functions. For decades, scientists believed this process ended with terminally differentiated cells. Committed to a particular cell fate, the ability to switch function irreversibly lost. In a major breakthrough, iPSC technology established the possibility of inducing plasticity to terminally differentiated cells. But several limitations arose, including a lengthy multi-step lab-based process, possible inefficiencies due to experimental conditions, low fidelity with which the final cell type is generated, and safety concerns, including potential cancer risk. More recently, scientists found that mature, fully differentiated somatic cells could be directly converted to another cell type by ectopic expression of specific transcription factors, without the need to acquire intermediate pluripotency. However, this technique requires viral gene transfer, an approach which, 1, requires an exhaustive laboratory procedure, and 2, carries risks, such as insertional mutagenesis, irreversibility caused by stable transduction, and increased cancer risk caused by long-term proto-oncogene expression. Therefore, it is not well suited for point-of-care applications. Thus, a non-viral tissue nanotransfection approach was developed for direct cell conversion in vivo by Professor Chandan K. Sen in collaboration with Professor James Lee. This work has been published in Nature Nanotechnology in 2017. This approach is simple, does not require elaborate laboratory procedures, and can be used as a point-of-care approach. This approach consists of 2 components. First, a hardware component, or chip, fabricated for efficient delivery of cell-specific cargo in murine model. Manufacturing of this chip has been scaled up to fit dimensions relevant to human need. Such chip has been successfully tested in preclinical porcine studies. Second, the cargo itself, which consists of a combination of different reprogramming factors. Thus, by applying a highly intense and focused electric field through arrayed nanochannels, this device nanoperates the adjacent tissue cell membranes and electrophoretically drives reprogramming factors into the cells. This new nano-electroporation technology is distinctly different from bulk electroporation. The simplicity and utility of this approach was demonstrated by nano-electroporating the skin that served as an agricultural land to cultivate cells of different functional abilities. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to endothelial cells that ultimately formed blood vessels with 7 days. These blood vessel networks anastomosed with the parent circulatory system and restored tissue and limb perfusion in 2 murine models of injury-induced ischemia. Similarly, using a different cargo, skin fibroblasts were converted to neuronal cells that showed similar gene expression profile and electrophysiological function to that of cells in the developing brain. This tissue nanotransfection platform technology has the potential to ultimately enable the use of patients' own tissue as a prolific immunosurveilled bioreactor to produce autologous cells that can resolve conditions locally or distally upon harvesting. This simple-to-implement nano-electroporation technology generates favorable biological responses. It is a one-time treatment applied topically and only lasts for a fraction of a second, without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene, protein, and drug delivery.
So this work was first reported in 2017, and Daniel and Durba and Shubhadeep. Durba is actually now in IIT Ropar, and this chip is now in preparation for human testing in Mumbai under the oversight of AIIMS New Delhi. So a lot of progress in the meanwhile has taken place. There were a couple of editorials in JAMA, Science Translational Medicine, so on and so forth, quite favorable. So people are very excited that you don't need fancy laboratories. You can actually use the body to reprogram a part of the tissue that can be converted to cell types that are of therapeutic interest and can be transplanted within the body, depending on the need. So what is the technology? The technology, most of you may have heard about bulk electroporation, where you electroporate and deliver gene that is shown on the top. What happens there is most of the cells, many of the cells die. All of the cells have a hole in their membrane, and then the gene is plasmids are left out, and they enter the cell, and the cells that were able to repair their hole, you consider that as gene delivery. But what happens is that when you do that type of injury to the cytoskeleton, you blunt inherent plasticity of that cell. So what you see at the bottom is nanoelectroporation, where we open up less than 2% of the total surface area of the cell. And using that approach, we now deliver, as you see to the left, this type of cargo. And by the way, depending on what we want to reprogram to whatever fate or state, the cargo changes, but the device remains the same. Now, more recently, we manufactured a more large-scale version of this chip that is, a high-scale manufacturing, industry design, and this was published in Nature Protocol. And here is a small video explaining what this chip actually does.
Certain organs in our body can serve as a bioreactor. Through real-time feedback from our immune system, we can grow new body parts. We can even grow them in one place and then transplant them to where they are needed. One well-suited organ for such bioreactor function is the skin. This largest organ of the body provides ample real estate to grow therapeutic functional tissue structures. Tissue nanotransfection technology, or TNT, enables in vivo reprogramming of the skin to produce functioning blood vessels, active nerve tissue, insulogenic tissue, and more. This process depends on specialized chips created from silicon wafers. A coating is added to the top, then lasers are used to write small holes into the chip, then etched deep into the wafer. On the other side, a new coating is added and then lithographed to create hollow microneedles. The chip is then etched to make the needles longer, cleaned, and then treated with a new coating to improve precision. The exact nanofabrication process for manufacturing the TNT chip is now reported in a new publication in Nature Protocol. The delivery mechanism consists of two components. First, the chip, fabricated for efficient delivery of cell-specific cargo. Second, the cargo itself, which consists of a combination of different reprogramming factors. The cargo is different for different applications. By applying a highly intense and focused electric field through arrayed nanochannels, this device nanoperates the abutting tissue cell membranes and electrophoretically drives reprogramming factors into the tissue. In this approach, the skin may be viewed as an agricultural land that can be used to cultivate cells and multicellular structures of different functional abilities, such as perfused blood vessels, electrophysiologically active nerve tissue, etc. Using a specific cargo, a combination of different reprogramming factors, skin fibroblasts were successfully converted to vasculogenic cells that ultimately formed perfused blood vessels within 7 days. These blood vessel networks anastomosed with the parent circulatory system and rescued against ischemic insults. Similarly, using a different cargo, neurogenic as well as neurotropic reprogramming skin cells were achieved. Such intervention proves to be promising to rescue against stroke as well as diabetic neuropathy. In future, TNT is expected to involve dispensable chips with injectable factors supplied as liquid-loaded cartridges. During use, the chips will be stamped on the skin. Such approach will be compatible with point-of-care using minimum training for local and remote use. This TNT platform technology has the potential to ultimately enable the use of the patient's own tissue as a prolific immunosurveilled bioreactor to produce tissue, grow body parts, and resolve conditions. It is a one-time treatment applied topically, only lasts for a fraction of a second, and can be applied at point of care without elaborate laboratory procedures. And it could have applications beyond reprogramming-based cell therapies, such as gene therapy, protein delivery, drug delivery, and cell-specific exosome labeling.
So now that you have an idea of what the technology is about, I'm going to share with you a few examples of what have been accomplished so far. So the first example that we already have published is that we have developed a particular cocktail, gene cocktail, which when TNT, so we call that the vasculogenic TNT, and Durva, Shubhadeep, and Kanaya are the primary drivers of that, and this part is already published in Nature Nano. And what we have been able to show, several experiments were done. This is one where the femoral artery has been transected. And once you transect the femoral artery, you have this type of a sort of auto amputation of the limb, severe ischemia followed by auto amputation. And what you see to the right is a leg that does not have the femoral and was subjected to a one-time 0.1 second TNT with our vasculogenic cocktail, and we show tens and thousands of new blood vessels that grow and rescue that leg. All of this work obviously is now published. I'm going to take you through the wide range of what have been accomplished so that you get a feel for what is possible, and maybe some of you will be interested in this. More recently, we took the vasculogenic cocktail, and this paper came out, I think in 2020, showing that in a sciatic nerve crush model, you could actually improve recovery by doing vasculogenic TNT. Now, this EFF that is shown here is E2B2, FLI1, and FOXC2, which is basically the trifecta vasculogenic cocktail that we use for TNT. The work by Savita and others in the group has achieved neurogenic and neurotrophic TNT. Let me explain what that is. So what you do is when you do that TNT, so we use in this case ABM, ASCL1, BRN2, MIT1L, as you see on the top. And in about 2 to 3 days, you start seeing the delivery, of course, of these genes into the fibroblast. In about 5 days, you start seeing neuron-specific markers. So this is a neuron-specific class 3 beta tubulin, TUJ1. Then you also have microtubule-associated protein, or MAP2. These start showing up. And in about day 20, about 3 weeks, you start seeing, if you do patch clamp, you start seeing electrophysiology. And it is also around that time that you also see, neurofilaments, you see synapsin, and this is now you're going longer, you see a choline acetyltransferase. These are all sort of in a temporal manner seen over time. And in vivo, when we actually patch the part of the skin, we were able to show excitable neurons, thousands of them that were reprogrammed. Then we harvested those neurons growing in the skin, put them back in the stroke-affected damaged brain, and were able to rescue brain function. So all of this is already published. Now, interestingly, when you do this type of neurogenic reprogramming, what happens is, of course, many fibroblasts of the skin become neural cells, but then the fibroblasts that do not become neural cells start making large amounts of, say, NGF, neurotrophin. So you have a very neurotrophic environment in the skin. And this neurotrophic environment of the skin can be leveraged to rescue diabetic polyneuropathy. With diabetes, these types of peripheral nerves start dying off, and this TNT-based neurotrophic environment of the skin helps rescue those. The next example just got published, I think, last month or so in Nature Regenerative Medicine, where we have been working on a model, because of the interest of the US military, who became very interested in this. We actually just reported a myogenic TNT, and this myogenic TNT was in a model of volumetric muscle loss of the tibialis anterior. This is in PubMed, just came, I think, a couple of weeks ago. And what we showed here is not only that you have myogenesis, but you have improved torque of the muscle. So basically, this is the functional recovery of the muscle is pretty effective. And together with the functional recovery, you have a bigger muscle. So the weight of the muscle is also higher. What Kanaya published a few weeks ago in the Journal of Clinical Investigation is that he studied a whole bunch of patients and showed that in a chronic wound patients, you have certain genetic pathways that are epigenetically silenced. So he used TNT in a new way. So what did he do? He used TNT in vivo to do cell-specific gene editing. So the first example of in vivo cell-specific gene editing was achieved in this paper that came out in JCI in July of 2022, a few months ago. This paper is also out, and I would encourage you to take a look. In another example, in a paper that just got published today in Molecular Therapy, another very well-known journal, this is Professor Gail Gordillo, our Chief of Plastic Surgery, and she is very interested in the problem of hemangioma. Now, if you take this, we have a few plastic surgeons here, so they will be very familiar with vascular malformations, and you see this in children. So if you take these cells and you inject them in mice, what happens is you get a tumor because of which the mouse dies in about 2 weeks. So what we did was we demonstrated that you can now find out what is the molecular mechanism of this tumorigenesis, and then you can target that. In this case, the target was MIR-126, which is an angiomere. So we delivered anti-angiomere using TNT. Mind you, again, one application, 0.1 second. That's all it takes. No second application. And based on that, we see remarkable, as you see with the blue line, all these animals are dead 100% of them are dead in 2 weeks. And you see in red, a lot of survival. Also to the right, you see survival and continue. We then followed up for 6 months, and we are able to see tumor-free survival. Dr. Ghatak, who is currently an associate professor in the group, what he did is that he used TNT to label exosomes fluorescently. So you know these exosomes are nano carriers of gene, protein, and many other factors. And what Subhadeep did was to see that in the blood, to be able to trace the origin of the exosome, he reported a technology that just got reported in ACS Nano, a highly reputed journal again, where he was able to demonstrate that exosomes originating from the keratinocytes actually are involved in keratinocyte macrophage crosstalk. And when this crosstalk happens, that is central to the overall process of wound healing. And if there is an impairment of that process, as is now seen in the cases of diabetes, once you know what this mechanism is, you can target that and you can correct that. Finally, Al Hasnain, one of my mentees, is a plastic surgeon. And Al just won for this project a major national award here in the US. So he is interested in lymphedema, especially after mastectomy. So he developed a model of a mouse tail vein lymphedema. And essentially, once you have this lymphedema, what the surgeons do is they do lymphovenous anastomosis. So they surgically do lymphovenous anastomosis. Now, interestingly, in developmental biology, the lymph and the lymphatic vessel and the vein is one structure. Eventually, it cleaves off. And there's a molecular process that cleaves it off. So what we were able to find is that using a particular molecular target where TNT was used for the delivery, we are able to reopen that or undo that and fuse the two without having to do any surgery. So this paper, one part of it just got published, and the bigger paper is currently in review. And Al is able to report a significant, as you will see with the red line, the black line versus the red line to your right, bottom right, a remarkable improvement. And again, it says TNT-LC. This is the lymphovenous anastomosis cocktail. So it's the LC. Of course, this is subject to patent, so it is not named exactly what it is. And finally, diabetic wound healing is obviously a major problem. And for most of you that know, and I just mentioned a short while ago, that once you have a wound in the fetus, it repairs perfectly. We have regenerative repair under fetal conditions. So we studied the feti to understand how that happens. And then in this case, we actually turned on that fetal pathway in the diabetic adult. and was able to improve wound healing. And this also was done using TNT. So the paper, this also came out just recently. So it's supposed to be out in January 2023, but the paper is already in PubMed. You should be able to read it. Driving adult tissue repair, especially diabetic adult tissue repair, where re-engagement of a pathway required for fetal healing. With that, I'd like to sort of conclude my talk here, so we have some time for discussion. Thank you very much for your attention. These are the sponsors, primarily the NIH and the US Department of Defense. Thank you.
Thank you very much, sir. It was really eye-opening and excellent. The amount of work being done under your mentorship, and I really congratulate you and your team and the center, and hope that you will be, and you are already into our Indian platform. And as a plastic surgeon, I wish to invite you in JIPMER Pondicherry Institute of National Importance, where we have our anti-aging and regenerative clinic since 2012. We are also doing a regenerative, but the level which you have achieved is tremendous. So we will love to hear from you. Because of the paucity of time, maybe we can take 1 or 2 questions if any audience has. If there is a no question, I would like to just ask one question, sir. Any question? Any question?
Yeah, we have a question in the chat box.
We have a question, sir.
Okay.
Yeah.
Sir, it is share your insights on long-term structural and functional behavior of tissue, nanotransfection tissue.
Yeah, very good question. So you see, whenever you do something like this, you're always concerned about safety. So far we have done 6-month follow-up studies, and we have also done a genome-wide mutagenesis study, based on which, so far, as you know, the alternative is viral. Once you do viral gene transfer, there is inflammation, and you have unrestricted propagation of the gene transfer potentially. Because this is a biophysical approach, you're using electric field to nanopurate. This does not propagate. So far, in the study that we have done up to 6 months on mutagenesis, we have not seen anything, and we have done whole body, tumorigenesis, pathology. So no negative, indications yet. But of course, this will have to be tested in humans. So far, rodents, pigs, and monkey done. So therefore, we have, we're approaching the human.
Thank you very much, sir. I think we should call it a day for this session. Why? Because the next program is also very important to the institute. And thank you very much, all the participants, and Dr. Sen. Thank you very much.
Thank you.
Dr. Koshi, do you have any questions?
Yes, if there is time, I have a quick question.
Yes, please go ahead.
Yes, Dr. Chandan Sen, thank you very much for this amazing keynote address. We in AVMC, we are indeed lucky to have had you. Yes, my question is, it's mind-boggling, the TNT technology, but what is it that makes the skin fibroblast have more plasticity than, say, the other cells, say a kidney cell or a bone cell? What do you think is the fundamental characteristics that makes you work on the skin fibroblast?
So first and foremost, the skin is much more robust in repair and regeneration in general, historical studies would show. But the specific answer to your question lies in the observation that the skin fibroblast is actually made up of 8 different types of cells. So when you do single cell sequencing, it is made up of 8 different types of cells, of which 5 clusters are housekeeping, and they are not plastic. But you have 3 clusters that are dedicated to plasticity. So whatever challenge you do, these 3 clusters can either move and become a neuron. Just injury itself, you don't have to reprogram. Just injury itself is a physiological signal that triggers reprogramming in these subclusters. So single cell sequencing is showing us that you have dedicated cells that account for this plasticity in the skin.
Thank you. Thank you.
Thank you.
Without much further delay, there is no other questions. We can end this session. And we'd like to thank our keynote speaker by giving a certificate of appreciation.
Thank you.
We'd like to thank our chairperson, Dr. Ravi Kumar Chitoria. Thank you, sir.
Thank you for giving me this opportunity.
We'd like to thank our next chairperson, Dr. Apen Koshi. Thank you, sir. Thank you for chairing our session.
Over to the organizers.
Thank you, sir. Thank you very much. Thank you so much, Dr. Chandan Sen, for a very insightful keynote address.
Thank you, madam. I appreciate
Thank
that.
you. Thank you, sir. Thank you very much.
Thank you, everyone. We'll now move on to our inaugural program. I once again wish everyone a very happy morning. On behalf of Arupadi Medical College and Hospital and its entire fraternity, with immense pleasure, we welcome you to BMSCON 2022, the third International E-Conference on Role of Basic Medical Sciences in Academia, Diagnosis and Research Advancement, organized by the preclinical departments. Organized by the preclinical departments of Anatomy, Physiology and Biochemistry, along with the Center for Biomedical Research, Arupadi Medical College. I, Dr. Aarti, will be emceeing this inaugural program. We'll just hold on for 2 minutes before we start the sessions. We welcome everyone back to BMSCON 2022. Now that our dignitaries have joined us, let's begin the conference. On behalf of Arupadi Medical College and Hospital and its entire fraternity, with immense pleasure, we welcome you all to BMSCON 2022. This is the third International E-Conference on the role of basic medical sciences in academia, diagnosis, and research advancement. It's organized by the preclinical departments of Anatomy, Physiology, Biochemistry, along with the Center for Biomedical Research. The purpose of this conference is to bring together clinicians, educationists, diagnostic consultants, and biomedical researchers on a single platform to share their knowledge towards the advancement of basic medical sciences. So let's begin by invoking the blessings of the Almighty with the Tamil Thai Varthi.
எக்கணமும் அதிசிறந்த திராவிடனல் திருநாடும்
Thank you. I request the organizing secretary, Dr. Lakshmi Jatia, Professor and Head Department of Physiology, to welcome the gathering.
Thank you, Dr. Aarti. Distinguished Chief Guest, Dr. Chandan Sen, Honorable Chancellor, Dr. A.S. Ganeshan, Sir, Mr. Suresh Samvil, Member, Board of Management, Dr. P.F. Couture, our Dean, Dr. Mahalakshmi, Dean HPE, Dr. Vishnu Bhatt, Director Medical Research, Dr. Jay Singh, Medical Superintendent, Dr. T. Rajan, Vice Principal, Dr. Anachalam, Vice Principal Student Affairs, Dr. Deepa, and Dr. B. Jayakumar, our DMS. Speakers, chairpersons, delegates, good morning and a very warm welcome to Galaxy of Luminaries. As organizing secretary, I, on behalf of the organizing team, take pride in welcoming you all to the inaugural ceremony of BMSCON 2022. We at Arupade Vidu Medical College and Hospital are honored and delighted to host this international conference for the third consecutive year. The theme of this conference is Role of Basic Medical Sciences in Academia, Diagnosis, and Research Advancement. This conference has the distinction of bringing together such a large number of resource faculty and delegates across the globe for a vibrant and intense exchange of views, knowledge, and experience on emerging trends in research, diagnosis, and innovative teaching learning methods. BMSCON has now become synonymous with Arupade Vidu Medical College. This year too, we had an overwhelming response with 800 registrations and 230 plus abstracts for Young Research Scholar Awards and oral presentations. This conference is the result of collaborative and synergistic efforts of the departments of anatomy, biochemistry, physiology, and Center for Biomedical Research. On this momentous occasion, we are honored to welcome our Honorable Chancellor, Dr. A.S. Ganeshan, Sir, a visionary who has been constantly supporting us in all our endeavors. Sir, I extend a warm welcome to you on behalf of the organizing committee and all of us present here today. Welcome, sir. The organizing team also welcomes our Honorable Chancellor, Ma'am, Dr. Anuradha Ganeshan, whose invaluable guidance is cherished by us all. Though Madam is not able to join us today, we welcome Madam and we seek her blessings for this program. The organizing committee is extremely delighted to welcome Dr. Chandan Sen, chief guest for today's inaugural program. He is an academician and ardent researcher working at Indiana University. We extend a warm welcome to you, sir. The organizing team also takes pride in welcoming Mr. Suresh Samvil, member, Board of Management, a man who is highly ambitious and lives by principles. Welcome, sir. We cordially welcome our Dean, Dr. P.F. Couture. He is a man with abated energy and enthusiasm. Welcome, sir. We also welcome Dr. T. Rajan, Vice Principal, who had been a guiding spirit and a crucial link between the organizing team and the management. We also welcome all the honored guests present here today, Dr. Mahalakshmi, Dean HPE, Dr. Jay Singh, Medical Superintendent, Dr. Vishnu Bhatt, Director Medical Research, Deans of other medical colleges under VMRF, university officials, members of STB, faculty members of AVMC, and friends from press and media on this auspicious occasion. We welcome delegates who are joining us from far and wide. Without your support and encouragement, this conference would not have been possible. We sincerely hope you will enjoy today and the next 2 days of deliberations and networking. I once again welcome you all, especially our Chancellor, Sir, Madam Chancellor, Dr. Chandan Sen, Mr. Suresh Samvil, other dignitaries and delegates to the inaugural ceremony of BMSCON. Thank you.
Thank you, ma'am. I now request Dr. Rajan T., Vice Principal, AVMC, to enlighten the gathering on the history and growth of BMSCON.
Thank you, Dr. Argani. Aarti, I'm audible?
Yes,
Yes,
sir.
sir. Audible, sir.
my screen is visible?
Yes, sir.
Yeah. Dignitaries on the screen and off the screen, dear delegates and the faculty of AVMC, a pleasant morning to all. With the support and divine blessings of God Almighty, our founder chairman, Dr. A. Sanmusundaram sir, and the benevolence of our trustee, Srimati Annapurni ma'am, in the year 2023, state conference was jointly organized by Dr. K.R. Srinivasan with Association of Anatomists Tamil Nadu, the Department of Anatomy of Harvard Medical College, Vinaya Missions Research Foundations, which made very good impact among the delegates. In the year 2007, the state conference has been again organized in the Department of Anatomy by Dr. K.R. Srinivasan, along with the Association of Anatomists Tamil Nadu, which created a very good image on Harvard Medical College, Vinaya Mission Research Foundations to conduct the conference. After 10 years, on one fine day, during the department meeting, it was decided to conduct an international conference in physical mode. It was organized during the year 2008 at Hotel Accord with the support of all the faculty in the Department of Anatomy. It was organized very well in a grand manner. Let me go for the growth of the BMSCON, how it is changed into from the ANATCON to the BMSCON. On the occasions, I would like to express my sincere thanks to each and everyone for the success of the first Inaugural conference. By seeing the grand success and better outcome of the conference, we decided to conduct the same in the next year. During the discussion, we decided to include the preclinical subject, anatomy, physiology, and biochemistry combinedly, as well as Center for Biomedical Research. We have discussed with other preclinical department HODs and faculties, along with the head of the Center for Biomedical Research. Everybody appreciated and accepted the good initiatives. With the support of Honorable Chancellor Dr. A.S. Ganesan sir, Director Dr. Anuradha Ganeshan, and guidance of Mr. Suresh Amal sir, Member Board of Management, and Dr. P.K. Sudhir sir, Vice Chancellor, and Dean Dr. P.F. Kotur, and the leadership team of the AVMC, it was conducted successfully in the November 2020. It's the first BMS conference. It was planned as a physical conference, but it was organized well in a virtual mode due to COVID. By seeing the appreciation and very good feedback from the delegates, it was decided to continue every year and conducted during the 2021. Around 10,000 participants were from the various countries. Now we back with the bank, and we are now at the virtual auditorium for the inaugural program of BMSCON 2022. BMSCON 2023 will be conducted hopefully in physical mode at our campus. I thank the organizing team for giving me the chance to share the history and growth of the BMSCON. Thank you, Dr. Arti.
Thank you, sir. I now request the organizing chairperson, Dr. M. Manju, Professor and Head, Department of Biochemistry, to address the gathering on the theme of this conference.
Thank you, Aarti. Respected dignitaries, esteemed speakers, chairpersons, and dear delegates, a very good morning. As the organizing chairperson of this third international conference, BMSCON 22, it's my honor and privilege to introduce the theme of the conference. The theme of this conference is Role of Basic Medical Sciences in Academia, Diagnosis, and Research Advancement. The theme for this conference was decided keeping in mind the major roles played by a doctor in a medical college, like academician, diagnosing and treating physician, and researcher, with special emphasis on recent advances on all these three fields. The medical professionals They should be aware of the latest research advances related to the basic medical sciences, as it helps to translate the bedside research into bedside management and be assertive in evidence-based practices for efficient and effective patient care. So keeping in mind, the topics were decided by the speakers, and the topics which will be covered in the sessions in these coming 3 days of the conference include role of skills, technology, and basic medical sciences in medical education, advanced research related to epigenetics, next generation sequencing, next generation cytogenomics, role of artificial intelligence in diagnosis, latest imaging technology in diagnosis, 3D printing in research, T-cell vaccines, immunotherapy, synthetic biology, gut microflora in health, and in vitro and in vivo studies related to latest scientific developments. Basic medical sciences. It forms the strong foundation for medicine and surgery and its allied specialties to build up. After the new CBME curriculum has been implemented into the medical colleges all over India in 2019, the teaching learning methods like early clinical exposure, self-directed learning, integrated teaching, small group teaching, skills training, DOP sessions, etc., they have gained prime importance. And diagnostic modalities in a lab testing related to clinical biochemistry, immunology, and molecular biology, they have advanced in leaps in the last few years. Information on such recent advancements in diagnostics helps the medical fraternity to render better patient care services. We hope this conference can serve as a wonderful platform for educationists, diagnostic consultants, and researchers to share their experiences and findings, which would be of great help for many other participants to gather, improvise, and put into practice the novel and innovative ideas. And I'm wishing you all a very wonderful scientific and academic BMSCON 2022. Thank you.
Thank you, ma'am. Light symbolizes knowledge and wisdom. To solemnize this special occasion, on behalf of the dignitaries, may we illuminate the lamp virtually. Thank you. May I now request our Honorable Chancellor VMRF, Dr. A.S. Ganesan, who is the epitome of excellent leadership, to address this gathering.
Very good morning to all the participants for this BMSCON 2022. It is my pleasure to be part of this inaugural session of this third international conference of basic medical sciences in academia, diagnostics, and research advancement. ABMC has been like has been trending this conferences, organizing these conferences since 2020. The first international conference was conducted in 2020 that emphasized on emerging trends in research in the field of basic medical sciences. The second international conference in 2021 focused mainly on exploring new modalities in teaching, learning, and research. And of course, the third one being in academia, diagnosis, and research advancement. The objectives of this conference, as you all know, is to enlighten the participants on the role of basic medical sciences in academia, diagnosis, and research in the field of medicine, and to serve as a platform for sharing the latest developments in the field of diagnosis, research, and teaching learning methodology related to basic medical sciences. And of course, to encourage young budding medical professionals to present their research work and update their knowledge, ultimately enhancing their network. I'm glad to note that faculty, PhD scholars, postgraduates and undergraduate of disciplines from medicine, dentistry, veterinary, nursing, physiotherapy, and allied health sciences are participating in this conference. And I'm sure that this conference is going to be extremely useful for all the participants to improve their knowledge and put into practice the novel and innovative ideas that they will gather from this conference. The key highlights of this conference being the number of registrations that has crossed 700, and the number of abstracts received for oral presentation about 235, and the number of Young Research Scholar Awards for UG, PG, faculty, and PhD categories contestants, with the cash prize and certificate. And the number of e-posters for UG is about 46. And I'm glad to know that we have about 7 international speakers and 9 from across the country. And I would like to thank all the participants, all the speakers who have taken the time and effort to share the knowledge for all the participants in this conference. And I would like to congratulate the advisors for this conference, Dr. Vishnu Bhatt, our Director of Medical Research, our Dean, Dr. Kotur, Dr. Mahalakshmi, Dr. Rajan, and the organizing committee chairperson, Dr. Manju, Professor Head Department of Biochemistry, Dr. Ilankadir, Dr. Lakshmi Jatia, Dr. Ravi Kumar, and the various committees that have taken all the effort in organizing this conference. I wish all the participants a grand success. Thank you.
Thank you very much, sir, for your wishes. It is with great enthusiasm that we have prepared the souvenir this year. It's a conglomeration of scientific knowledge and the various aspects of this conference. I now request our respected member BOM, Suresh Samuel, sir, to release the souvenir.
Thank you, Dr. Arati. Good morning, all of you. It's my pleasure and honor to release the souvenir for the 3rd International E-Conference on role of basic medical sciences in academia, diagnosis, and research advancement.
Thank you, sir. May I now request our member BOM to deliver the felicitation address.
Honorable Chancellor, Dr. A.S. Ganeshan, respected dignitaries, online, offline, participants, online, offline, Team AVMC, the organizing team, Dr. Chandan K. Sen, Chief Guest for this conference, the leadership under Dr. P.F. Kotur, Dr. Vishnu Bhatt, Dr. Mahalakshmi, Dr. Jai Singh. His hearty congratulations to the entire team in organizing this 3rd International Conference on basic medical sciences. In this juncture, I would like to also highlight one important area that what this conference will also be focusing upon. specifically with regard to medical informatics. I think medical informatics is the science and art of processing medical information. In this age of information explosion, choosing the useful one is rather difficult, and there lies the scope of electronic database management. However, still many outstanding personnel related to the healthcare sector take pride in being computer illiterate. The onus of the best use lies on the end user healthcare providers only. Another term, telehealth, encompasses all the e-health and telemedicine services. Computer aided or assisted learning is a computer-based tutorial method that uses the computer to pose questions, provide remedial information, and chart a student through a course. Now, the emphasis in medical education is on problem-based learning, and the computer aided or assisted learning could be of utmost help if used judiciously. Basic medical education and research lays the foundation for advancing and applying proper healthcare delivery system. There is no doubt that deep knowledge of anatomy is mandatory for successful surgery. Also, comprehensive knowledge of physiology is essential for grasping the principles of pathology and pharmacology adequately to avoid incorrect and inadequate practice of medicine. In this regard, the medical informatics is not just a subject to be learned and forgotten after the first professional MBBS examination, but this is something which would really help in terms of the artificial intelligence, health data management, health analytics, health information, which is how the medical programs and the medical care is being steered forward. So integrating the technology aspects in this basic medical science in today's conference is something very encouraging and in thing at this point of time. We are going to cover subjects with regard to 3D printing, with also to health informatics, with regard to health database management, and also extending it for newer technologies areas, how specifically we can look in terms of the healthcare adoption with regard to blockchain technology for more secure transactions and likewise. So I wish the conference a great success and happy to see this organizing committee has put together a very comprehensive agenda. I wish all the participants and also thank all the speakers for participating in this conference. Wish you all the very best.
Thank you very much, sir. We are very honored to have with us today our chief guest, Dr. Chandan K. Sen. Dr. Chandan K. Sen is a distinguished professor, J. Stanley Battersby Chair and Professor of Surgery, Director of Indiana Center for Regenerative Medicine and Engineering, Executive Director of the Comprehensive Wound Care Center at the Indiana University Methodist Hospital, and where he serves as Associate Vice President of Research and Associate Dean of Research at the School of Medicine, Indiana University, USA. His research is funded by prestigious agencies, US Department of Defense, US Department of Veteran Affairs, and the National Institutes of Health. He is also an elected member to the National Academy of Inventors. Sir, we are very happy to have you with us today. I now request you to address the gathering.
A very good morning to all dignitaries and teachers, students. This is quite a special experience for me to participate in an event like this and to see the enthusiasm of the institutional leaders and their commitment to develop research and education. It's clear that in our country of India, and obviously there is a large need, there is a large gap in healthcare that we must all fulfill. But together with that, as we do that, there comes other responsibilities that are integral. Such responsibilities include assuming global leadership in specific aspects of healthcare. That is the only way that we could generate cutting-edge solutions in a way that could be affordable for our citizens. There is a tremendous amount of science going on around the world, but unfortunately, not all of that is affordable. And in my opinion, the only path to affordable healthcare within this vast nation of ours is for us, some of us will be within the country, some of us will be outside the country. But together, we must assume leadership to a point where we can grow solutions within the country and disseminate that to people of all strata. To that end, I think the commitment of your institution, training your students, setting a curriculum that respects what we learn traditionally, but also is responsive to the changing foundations of science are critically important. I think through events like this, you would be able to achieve those changes. And some of us that are perhaps geographically not in proximity, but share a common interest, could pull together and help achieve what seems like a large task, but together, definitely it is achievable. I have utmost confidence in our new generation and their interest to assume global leadership. To that end, we must enable our students and must ensure that the resources that are needed for such aspiration are provided. I'm quite confident that the Institute leadership would cater to those needs. And I thank all of you for having me here, being a part of your family. Thanks to all of you.
Thank you very much, sir. I now request our beloved Dean ABMC, Professor P.F. Kottur, to deliver the presidential address.
A very good morning to one and all over here. Respected chief guest of this inaugural function and the keynote speaker, Dr. Chandan Sen, Honorable Chancellor of our prestigious Vinayaka Missions Research Foundation, deemed University, Dr. E.S. Ganeshan, our beloved member of the Board of Management, Mr. Suresh Samuel, and my colleagues, head of the institutions, other constituent units, and the organizing secretaries, Professor Lakshmi, Professor Manju, Professor Jalankati, my vice principal's colleagues, Dr. Arunachalam, Dr. Rajan, and the Director of Research, Professor Vishnu Bhatt, the staff members and invitees, resource persons and participants from across the country and across the globe. As a primary host and as a head of the institution of this virtual platform of AVIMC, I'm indeed honored and privileged to deliver this presidential remark. And an humble beginning, which was made 3 years back, when the COVID struck all of us, the humble beginning to drive away the claustrophobia has resulted in a short span of 3 years an institutional character of AVMC. This international conference has become a character and is going to be continued uninterruptedly. That's what is the commitment. And for this to happen, it is my prime duty to, of course, pay respect to our founder chancellor, Dr. A. Shanmugasundaram, Madam Shanmugasundaram, and our beloved Honorable Chancellor, Madam Chancellor Dr. Anuradha Ganeshan, and the esteemed Board of Management and the university at large, the Pro-Chancellors, Vice-Chancellors, and the University Administration, being proactive in promoting such events. I'm very proud to say that our management and the university at large have adopted an aggressive approach to promote research at all levels. And a lot of investments have been made in terms of equipment, in terms of human resource, in terms of incentives offered to the researchers. And there has been a lot of accelerated activities to promote research at all levels has happened. And now we all know this is an era of evidence-based medicine and personalized medicine, wherein every disease or ailment is being defined at the molecular level, at the biochemical level. And such research in this innovative areas, emerging areas, will answer many questions which remain unanswered for years together. And this environment, research environment that is prevailing over here in the AVMC or the VMRF campuses, certainly I consider the students, our students, very much lucky. I already said that a new curriculum has been rolled out, new methods, early cell exposure, early problem-based learning. We are offering our students early exposure to basic medical science research. I consider the students to be very, very lucky. And we all know that research and education are the two sides of the same coin and go hand in hand. And if we are laying down the foundation to create a new Indian medical graduate or a global doctor by inculcating or impregnating all these paradigms in the early part of their career, so that when the final product comes out as an AVMC defined product into the global platform, it is quite valued, appreciated. That's what is the perception as a teacher, as a head of institution, I foresee. And at this point of time, I must thank all the resource persons coming from different parts of the country, different parts of globe, and the participants from across, nearly 1,000 participants, and also the under students who have participated in this. And I'm sure they will be able to make most of this opportunity that is put forth in front of them. I must thank all the resource persons for having respected our request to share their expertise, to share their knowledge on this platform. And my special thanks to the chief guest, Dr. Chandan Sen, whose eye-opening key address, addressing many innovative discoveries he has shown us. So special thanks for him also. So once again, I thank and wish that the next 3 days the platform is going to offer quite a lot of food for thought and for the improvement of all the participants, staff, and students. Thank you very much. Namaste.
Thank you very much, sir. I now request Dr. S. Elankadir, Professor and Head, Department of Anatomy, to propose the vote of thanks.
Thank you, Dr. Aarti. Good morning, one and all. It is the biggest day in the history of our institute. Today, we have hosted the biggest academic conference in India. As an organizing chairperson, I really feel honored and privileged to get the opportunity to propose the vote of thanks on this special occasion, for which I humbly thank the organizing team BMSCON 22 for giving me the chance. I thank the Almighty for the blessings showered upon us for the ever success of our conference. I want to express my heartfelt gratitude to our founder chancellor, late Dr. A. Shanmugasundaram sir, and Mrs. Annapurni Shanmugasundaram madam, who have provided us this platform for conducting this mega conference. On behalf of our university, institute, and the organizing team, I would like to thank our honorable chief guest today, Dr. Chandan Sen, Director of Indiana Center for Regenerative Medicine and Engineering, USA, for his guest lecture. Sir, you have readily accepted our invitation and granted us your valuable time amidst your busy schedule to enlighten us in tissue reprogramming in regenerative medicine. It was a wonderful session, sir. Thank you. From the bottom of my heart, I would like to thank our beloved Honorable Chancellor,
Dr. A.S. Ganesan sir, who always welcome us whenever we try to approach him with any academic proposals. As a student, as a faculty, I have always thought how these many academic programs across various disciplines are being conducted in this institute. No one needs to tell me now. I have the answer. It's you, sir, who have been behind the success of all the academic programs conducted in our institute. Thank you, sir, for your valuable and gracious presence with us in this conference. I also would like to express my sincere thanks to our beloved Honorable Madam Chancellor, Dr. Anuradha Ganesan Madam, in absentia, who always stood beside sir and supported us in organizing our conference. Your wishes are always with us, Madam. Thank you, ma'am. My sincere thanks to our beloved Vice Chancellor, Dr. P.K. Sudhir sir, in absentia, who have always showed interest and supported us in conducting our conference. His wishes are always with us. I also take this opportunity to thank our other VMRF administrators for supporting us in organizing this mega event. On behalf of the organizing team, I would like to extend my sincere thanks to Member Board of Management, Mr. J. Suresh Samuel sir, for his constant encouragement and support in organizing this conference. Whenever we come across any hurdle, sir has always been a bridge and paved us the way for our smooth conduct of this program. I also take this opportunity to thank sir for releasing our e-souvenir and delivering the felicitation address. We thank you, sir. Deep from my heart, I would like to thank our beloved Dean, Dr. P.F. Kotu, sir, for his constant support. Really wondered, sir, you are equipped with enormous experience. Your support has given us a framework upon which we have planned and executed this program. Thank you, sir. We also thank you, sir, for delivering the presidential address today and for your constant support and inspiration. Now, I would like to put forward my sincere thanks to our advisory members, Director, Medical Research, Dr. Vishnu Bhatt, sir, Dean HPE, Dr. V.N. Mahalakshmi, ma'am, Medical Superintendent, Dr. Jay Singh, sir, Vice Principal Academics, Dr. T. Rajan, sir, Vice Principal Student Affairs, Dr. Arunachalam, sir, Deputy Medical Superintendents, Dr. Deepa and Dr. Jaykumar, for their constant support. Thank you all. I also would like to thank the heads and staff of our other colleges under VMRF for their valuable support. Thank you all. I also would like to express my heartfelt gratitude to our esteemed speakers, chairpersons, and judges who have consented to support us in this venture. Thank you. I also thank the entire team BMSCON 22 who have tirelessly worked for the conduct of the program. Upon my personal experience and from other administrators, faculties of our institute, wherever we go for attending any academic programs like conferences, CMEs, we have been constantly inquired by various sources like, when is your next conference BMSCON? That depicts our success in conducting the conference. If time is money, we have millions now added to our vault. It's time to thank you all enthusiastic delegates globally who have dedicated their valuable time by showing interest upon registering for our conference, participating live today, and making our conference a benchmark in history. Once again, I thank each and everyone present live in an ambition to make our conference a grand success. Thank you all. Thank you.
Thank you very much, sir. I thank all the dignitaries who have joined us today. We shall now conclude this inaugural program with the national anthem. I request everyone to raise for the national anthem.
जनगणमन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल गंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे।
Thank you all. We request everyone to stay logged in for the scientific sessions, which will begin right away. Thank you, everyone. And we also again thank the dignitaries for joining us today.
Dear delegates, we'll be starting our scientific session in another 2 minutes. Kindly stay on live.